Welcome back, friends. We are talking about the developmental biology, and we have seen the developmental biology of lower organisms. We have seen the developmental biology of sea urchin, developmental biology of chick, and also developmental biology of Drosophila. Now let us focus on a little bit about the developmental biology of human being. Now in this case, we will be talking about two important part of the developmental biology, which is called the spermatogenesis and also oogenesis. Now we'll be focusing on spermatogenesis and oogenesis separately uh, in different columns in this board. Then we will be connecting and we will be comparing the spermatogenesis with oogenesis, because I have found it very much interesting and also simple and easier to understand when we compare the spermatogenesis with oogenesis. Otherwise you may confuse two different things. Now another important thing about this video is that I am not going to talk about the anatomy of the female and male reproductive system. We'll be doing this in the separate video, but in this video I'll be talking about the cells and the maturation of cells during the spermatogenesis and also oogenesis, to give you an idea of how the cell maturation is going on from the uh, different developmental time. Now the developmental time we'll be taking here is, one is the before birth, then after birth till puberty, then we'll be talking about from the puberty till the death for the male, and also from the puberty till menopause for women. Now let's come here. So we'll be talking about spermatogenesis and oogenesis. So that means we are talking about gametogenesis. Okay, so we'll be talking about gametogenesis. Okay, so gametogenesis means in case of male and in case of female, the process of gametogenesis actually varies. Now we'll be dividing it into two different parts. We'll be talking about male here and we'll be talking about female here. Now the gametogenesis process for male is quite simpler than the gametogenesis for female, because the female gametogenesis or oogenesis process is complicated. Now female gametogenesis is called oogenesis and the male gametogenesis process is called spermatogenesis, because the name suggests it all, as genesis means the production or the generation of sperm, so we call it spermatogenesis. In case of oogenesis, again it's the genesis of oocytes. Oocytes are the mature cells. When oocytes are getting matured, it will produce the egg cells actually. Okay, so let's begin. Now in this case we will be looking at the examples in different way, like the cells and the modification of the cells to give rise to sperm and ovum. So whatever we are seeing, we have seen different names of the cells. Sometimes the cell is called oogonium, sometimes it will be called oocyte, and so on. So we will be seeing similarity between the naming of cells in case of spermatogenesis and oogenesis. Now let's begin with the spermatogenesis process. Now in this case what we can see, uh, again I have told you that we will be dividing this whole process in different timing segment, because remember in developmental biology it's nothing but some sequential events finely placed in times, and the place of the expression of certain important traits are important. So let's begin with here. So we'll be dividing into several parts. So this is say before birth. So I'll be dividing the first row before birth, then we'll be talking about from the birth to puberty, and then the third part we'll be talking about from the puberty to death. So here it is, so this is before birth, then from the birth to puberty, then from the puberty till death. This is for the male, but for the female only this part differs. Only from the female, in case of female it will be from the puberty to menopause. Okay, so now let us talk about the process of spermatogenesis in male. So spermatogenesis actually is done inside the male, and there are cells, specialized cells and specialized organ system are present in both the cases to supply nutrients to any kind of gametes. In case of male and female, both the case, we have seen the different cells and uh, total system or organ system is produced in such a way to develop to give the nourishment to the developing gamete, and also nourishing those gamete and finally guide those gamete to establish a perfect fertilization event. Now we'll be seeing those anatomy. We'll be taking the names only, but we'll be not discussing the anatomy of them, because I've told you that we'll be doing separate video of those anatomical parts. Now in this case, the first kind of cells that are being produced before birth of both male and female are from the stem cells. Because remember, during the developmental stage of our life, from the embryo, so these are called the embryonic stem cells, and from those embryonic stem cells particular type of cells are generated in case of both male and female. Now those cells, so we are having the embryonic stem cells. So let me draw here. So this is embryonic stem cells. So ESC, it is designated as the embryonic stem cell here. From this embryonic stem cells, in case of male, what we've generated a cell which is called the primary, so it is called the oogonium. Actually it's called the spermatogonium in case of male, and in case of female it is called the oogonium. So spermatogonium is produced. So say this is the nucleus, this is called spermatogonium. Spermatogonia, and more spermatogonia will be called spermatogonium. So spermatogonium is a plural of spermatogonia. So many spermatogonia are produced, which we call spermatogonium. Okay. Now from the same type of embryonic stem cell, in case of female, we have seen the production of oogonium. So we have produced here oogonium. So oogonia, and from this we will produce oogonium. So this production of this spermatogonium and oogonium will happen before the birth of that particular baby. So it is going on during the developmental stages, after the gastrulation obviously. Now then this oogonium is having the capability. So these are the totipotent cell. Okay, so these kind of cells are actually totipotent cells. So they can produce many more cells. They can produce cells like themselves, and also they can produce cells like others. Now they are doing multiple multiplication via the mitotic cell division. So mitosis is going on both the cases. They start producing more and more oogonia, and soon they produce a channel and a cell line of oogonium in case of female. In case of male also, from the spermatogonia it will start dividing and produce many more spermatogonia. So it will produce more and more spermatogonia in this case. And in case of female also, we have seen they start go and differentiating and producing many more oogonium. But actually in case of females, the production of this oogonium is more than in case of males, because in case of males also there are production of spermatogonia, many spermatogonia, but those spermatogonia production is less than the production of oogonium. Now what these oogoniums are producing, after the production of this more oogonium, some of the oogonium will be converted. Some of the oogonia is converted into a mature kind of cell, and that mature cell is called primary oocyte. They are called primary oocyte. Now this primary oocyte is having a particular chromosomal characteristic and also everything needed for the proper cell division. So what they produce primary oocyte. So here, so let me draw this. So with this black dot or black nucleus, whatever I am drawing, you can say these are primary oocyte. From the oogonium they produce primary oocyte. We call it primary oocyte. Okay. Now in case of male, what will happen? This spermatogonia will start dividing and maturing into primary spermatocyte. Now the primary spermatocyte. So we have production of many more cells called primary spermatocyte. Now after the birth, even after the birth, in case of this male, in case of males, this spermatogonia will be converted into primary spermatocytes. So we'll be calling them primary spermatocyte. Now this primary spermatocyte will be then further matured into secondary spermatocyte, and from these spermatocytes, so sorry, not secondary, from the spermatocyte, after the maturation of the spermatocyte, what they produce are different types of cells. The cells, when they reach their puberty, so this is the sequential events. Before the birth, what they produce, spermatogonia. After the birth, the spermatogonia will be matured into primary spermatocyte. Now all of these are primary spermatocyte. With this green thing, we call them primary spermatocyte. Now as the primary spermatocyte will be getting matured, when they are getting matured, when they receive some hormonal signal. Now the hormonal signal they need for the maturation of spermatocyte into another specialized cells will be needed, and those hormones are testosterone. Now during the puberty of male, they receives enormous amount of testosterone. So as a result of this testosterone stimulation, these primary spermatocytes will be matured into what we call spermatids. So we'll be looking at the cells like this. They will be called spermatids. So these are spermatids. But the production of the spermatid from primary spermatocyte will only happen when they receive the signal from testosterone during puberty. So this is a very important part. After the production of spermatids, the spermatid will be matured and they will produce what we call sperms, right? And the production of this sperm, the capacity or capability of producing this sperm is throughout the lifetime for a man, right? Why? Because throughout the lifetime they are producing this primary spermatocyte, and we know after the puberty, anytime if they are stimulated with more and more testosterone hormones and hormonal signals, this primary spermatocyte will be matured into spermatids, and then from the spermatids it will be producing sperms. That is the actual thing. Okay, so this is the whole picture in case of male. Now in case of female, what will happen? Before the birth, we have talked about the similarity. Oogonia will be matured into primary oocyte, but after the production of primary oocyte, they will start meiosis division. They will start meiotic division. But the production of this, so this primary oocyte generation is a very, very important part of this whole developmental phase of female gametogenesis. Because in case of female gametogenesis, they haven't produced any kind of mature egg cell before birth. So what they produce? All this spermatocyte. Remember, in case of this male reproductive system, they start producing spermatogonia throughout the time. So throughout their lifetime, they can produce spermatogonia. So we know the spermatogonia are the earlier type of cells. So if we produce spermatogonia, we can produce primary spermatocyte, we can produce spermatids, we can produce sperm. So that's why in case of males, they are having the capability of producing sperms throughout the body time, so throughout the lifetime till death. That's why I have shown here till death. But in case of female, that never happens. In case of females, females lose the capability of producing oogonia after the birth. Okay, so they can only produce oogonia only before the birth, but after the birth they cannot produce any more oogonia. So the primary oocyte that is developed before the birth, that line of primary oocyte is everything, is every treasure for a female reproductive system, because after the birth no further oogonium will be produced, no further primary spermatocyte will be primary oocyte will be produced, because after this birth, this primary oocyte will have a mitotic arrest. This will lead to a mitotic arrest. It will receive the mitotic arrest at prophase 1. So mitotic arrest at prophase 1 will restrict the further production of primary oocyte. So the primary oocyte that is being produced before birth is everything for a female to continue and contribute to this reproductive system. So this is very important and unique feature which differing the spermatogenesis and oogenesis for the first case. Now let us consider the future steps. Now we know that after the birth, what we have in the baby female, that we are having primary oocyte pool inside the body, and that is everything, because this primary oocyte pool is arrested at its prophase 1, so they won't divide further. Now what we will see now they will start grow and mature. Now as they start grow and mature, when they reaches the puberty, they start receiving signals. Signals like, so some of this primary oocyte will start to mature, and they start to mature into secondary oocyte. Okay, so they will start mature into secondary oocyte. So no further primary spermatocyte will produce. Instead, they start dividing in a meiosis way, and they will start producing primary oocyte. Now as they are producing secondary oocyte, so they will producing secondary oocyte. So these are secondary oocyte. These are primary. Now these are secondary oocyte. Okay, so secondary oocyte. Now how they produce secondary oocyte? So we are having this primary oocyte which is having the chromosomes. For example, this is the chromosome, these are the chromosomes. Now what will do, it will divide in such a way in asymmetric division. Now what do you mean by asymmetric division? That they will divide in such a way that one of the cell receive more cytoplasm than another, and they will receive most of these chromosomes, and here the other will receive fewer amount of chromosome. So this small cell which is produced from the primary oocyte eventually getting degradation. Now this cell is called polar body. Now the polar bodies are produced during the oocyte, during the secondary oocyte formation. They will be degraded, and those polar body are not required, and those polar body can be produced due to the asymmetric cell division of the primary oocyte. Okay, so this thing is important, so you must know this thing. Okay, now they produce this secondary oocyte from the primary oocyte, and they produce several polar bodies which will be degraded in turn, so we won't bother about the polar bodies. Now they produce the secondary oocyte. Few of the primary oocyte is converted into the secondary oocyte, and during the puberty of the female, what will happen, they again receive some hormonal signal. Now the hormonal signal females usually receives is for LH, or luteinizing hormone. Now this luteinizing hormone and also the hormones called FSH, or follicle stimulating hormones, play an important role in controlling the further future events, which are much more complex there than that of the male reproductive system. Now here we will be talking about the menstrual cycle. Now here what is going on, that the secondary oocyte is produced. Now it is ready for the further division. Now the secondary oocyte will start dividing, but they again encounter another meiosis arrest, and the arrest they encounter is in the second meiotic division. First meiosis division, in the meiosis, first meiotic division happens or occurs perfectly, but in the second meiotic division, metaphase 2, so in the metaphase 2 they encounter another arrest. So during the cell division in meiosis, they have encountered arrest at metaphase 2. So this secondary oocyte are arrested in the cell division at metaphase 2. That means they have successfully completed the first meiotic division of the meiosis, but in the second meiotic division at metaphase they are arrested. Now in this phase they will remain throughout the time until and unless they have encountered the attack by a sperm. This is the very important part. This is the regulatory part that regulates the further cell division for the secondary oocyte and production of a full egg. Only after the fertilization this secondary oocyte will produce an egg. Otherwise it won't produce an egg. Okay, so this is depending upon the fusion of sperm. The fusion of sperm to the secondary oocyte give the secondary oocyte a signal which will remove the cell arrest at metaphase 2, and the cell will move through the meiosis, and it will complete the meiosis and produce the zygote. Okay, so this is a very important step. Now at this point it receives signal from LH, luteinizing hormone. So as a result of that, these cells before the development, so during the development, all of these cells are in the clusters, and we call the clusters follicle. Now the follicles are very important. Now as they are moving, now all of these things are happening in the ovary. Now after doing all these things in the ovary, they start to move outwards to the fallopian tube or oviduct. It is also called the oviduct. Now as luteinizing hormone comes and signal all the cells, they receive the signal and it will tell it to go and released into the oviduct. So now they move on and follicle is released, and the cells of secondary oocyte will be released to the oviduct or released to the fallopian tube. Now they will migrate through the fallopian tube in search for the attachment of sperm. But if now there comes two different stages. So two possibilities are there. That one thing is that they encounter sperm, second thing is that there was no sperm. Now if there is no sperm which is encountering this secondary oocyte, after it is released in the fallopian tube, then what will have, they will receive no signal for the further division, so they remain in the cell arrested phase and they will go and be released from the body. They will be released from the body along with several different factors, along with some blood and pus and all these things they will release. But what will happen when they encounter the sperm, when they encounter it, what will happen, this will fuse, and we know that inside the sperm we are having the pronucleus. So again in number of chromosome having this pronucleus. So after it encounters here this secondary oocyte, then the sperm attachment to the secondary oocyte happens. It eventually gives the signal to secondary oocyte, and the cell cycle arrest at metaphase 2 just come out and shuts off, and then it will start dividing by the second round of meiosis, and then it will go and produce a zygote. So if it encounters sperm, so if this thing happens, what we have done, we have seen. But if it encounters sperm, then what will happen, this secondary oocyte will be attached with the nucleus from the sperm. So here is the nucleus number of n, here is the nucleus number of n. So the nuclear fusion will be happen, and after that it will produce a 2n number of chromosome containing a large cell which we call a zygote. And now the zygote will be ready to be implanted onto the wall of fallopian tubing, and there are some proteins are also ready at this time. So remember, not only we have to make ready all these things, but we need to make ready other proteins which will be needed if it encounters sperm. So in every time during the puberty until menopause inside a female body, in every month during a perfect particular time, the cells and all the system is getting ready to have a zygote implanted onto the vaginal wall, onto the wall of the fallopian tube. Okay, so they are always starting this onto the wall of vagina. They are always start to do these things. But if they encounter sperm, this thing happens perfectly, and then the production of embryogenesis will start and the women become pregnant. But if they haven't encountered sperm, then every process that till date it produces will be released through a secretion we call the menstrual fluid. In the menstrual fluid we'll be having the secondary oocytes, and also we'll be having all of this tissue debris that are being produced to hold on to the perfect structure for the implantation of that zygote. So this is the menstrual fluid, and this is the menstrual cycle which is getting repeatedly happening inside the woman body from the puberty until menopause. Okay, so this is the important fact that is going on. Okay, so that's why I have told you that the female reproductive system is much more complicated, and also the female spermatogenesis or oogenesis is much more complicated than spermatogenesis. Okay, because in spermatogenesis we have seen the simple development, because they are having the capability of producing primary spermatocytes throughout the time, primary spermatogonia throughout the time. So in any man can produce sperm throughout its lifetime until death. Okay, maybe the production capacity will be reduced, but they can produce. But in case of women, this will happen only until a particular age, which is called the menopause barrier for the woman. Okay, after that all of these things will be stopped, and all this process will be halted, because remember, the female is having only a certain pool of primary oocyte, because if they run out of primary oocyte, there are no cells to support the production of secondary oocyte, and if there is no cell to support the production of secondary oocyte, there is no way to produce and go through cell division and to produce the zygotes and do all the secondary stuffs that we are seeing. So this part is very, very important, and we have seen the differences between different layers of the spermatogenesis and oogenesis. Okay, so that's it, and I hope this video will help you. But in the future videos we'll be also talking about the different effects and how it is done, and combining the anatomical features of male and female reproductive system, and we'll be seeing the different processes. But the thing is again the same: spermatogonium or oogonium, from there to primary spermatocyte or primary oocyte, from the primary spermatocyte to spermatids and sperm, from the primary oocyte to the secondary oocyte, from the secondary oocyte it will be produced via asymmetric cell division, and the polar bodies are the small cells having less amount of chromosome and all these things. They will be degraded and they will produce a secondary oocyte. Now the secondary oocyte go and arrest into the metaphase 2, and they will resume the arrest only after encountering the sperm. Then the fusion of the nucleuses and production of zygote having 2n number of chromosomes happen, and then they will be ready to be implanted inside the uh, fallopian inside the vaginal wall, and they'll start grow and produce the embryo. So this thing is the whole difference between the spermatogenesis and oogenesis, and I hope this will help you. Thank you.